again, and today we're going to talk about photosynthesis as well as certain experiments, right, that were done to show that photosynthesis requires certain factors. Okay, so we all know that humans as well as other animals, we need to eat, right, in order to survive. And sometimes we even see certain carnivorous plants that digest insects. But most of the time we see plants, they do not eat, right, in the typical sense. So how do we know that, where do plants get their food from, right? How do you get their nutritional requirements? So we realize that in order to perform all these vital functions in the body, the organism always needs to have energy, right? And so where to get this energy from is by breaking down energy-rich organic molecules. Right? One example would be glucose, right? When we eat glucose, our body can break it down during respiration. So when we trace the origins of all these organic molecules, our glucose, our starch and everything, we will always find that they come from green plants. So we are having our breakfast. In fact, I'm having my breakfast right now. And so we have coffee, toast, hard-boiled egg. Right? Where do all these molecules come from? We can see that the corn, all right, is used to feed the chicken, right? And the chicken therefore has the energy to lay the egg, right? And then we can eat the egg, which consists of some protein and then some of the fats. And then the coffee plant, right? The coffee plant is able to do photosynthesis, and this is the coffee fruits, right? We use the coffee fruit, right? Especially the seed, coffee seed, and then we can make coffee. And so bread, right? Bread comes from the wheat plant. When we bake it, becomes bread. So indirectly or directly, always comes from the molecules always come from plants. Okay, so then this Dutch physician called John Baptist, right? He conducted an experiment. He was very curious, right? He wants to know, does the plant use up the soil? All right, does it use up something in the soil? So what he did, right? He took a young willow plant, right, that weighed had a mass of 2.25 kg, as well as the soil with a dry mass. Dry mass means no water, of 90 kg. And then he put inside, right? And then he give rainwater. So the lid covers the soil, prevent evaporation. Okay, now 5 years later, all right, and so he's taking the mass of the plant, right, of the entire plant with just the trunk, the roots, and the branches. He realized that okay, is now 76.1 kg. The soil in that case is barely changed, right? It has barely changed in terms of its mass, so it's just lost 0.1 kg. Then why? So why did the mass of the tree increase by so much, but the mass of the soil decreased by so little? So where must the plants be getting their food from? So he realized that okay, last time people thought that actually the plant is sucking up things from the soil lah. Okay, but it shows that this experiment shows that the plant does not actually get the food right from the soil. It actually gets it from the atmosphere. Right? So what does the soil contain? Yes, the soil contains two things: water, which it constantly needs to take in through right through the roots, and the water also contains mineral salts, but the soil does not contain the carbon compounds. Okay, so of course the question is, where do these carbon compounds come from? We all know the answer to that, right? The answer is that the carbon dioxide in the air. So that's one. So the next thing is, how about light, right? What is the role of light in photosynthesis? So we say that if carbon dioxide and light are the sources of carbon and energy for making organic molecules in plants, how is carbon dioxide in the air converted into large molecules such as carbohydrates, right? So carbon dioxide, CO2, is a very small, tiny molecule. How do we transform this carbon dioxide into glucose, right, which has 6 carbon molecules? And how is the energy in light transformed into the chemical energy stored in these organic molecules? So the answer, of course, is the process of photosynthesis. So long time ago, right, the Earth, right, they have no life, right? There was just a barren planet. But because of the plants and because of the photosynthetic bacteria and algae that are found in the ocean, therefore there's oxygen in the atmosphere, and that gives life on Earth, right? So all life depends on photosynthesis. So this is the process of photosynthesis. We call this as the word equation, A very important equation, you must remember. Okay, so when you're writing this in your exam, this is called the overall word equation. Please do not forget any single word, right? Including these two words over here. So on the left, we have carbon dioxide plus water, arrow, light energy on top of the arrow, chlorophyll below the arrow. Do not write chloroplast. Chloroplast is the organelle that contains the pigment chlorophyll. So here, below the arrow, we write chlorophyll. The products are glucose and oxygen. Okay, so the plant can use glucose, right, to convert to other substances, use it for photosynthesis, I use for energy. But the plant, yes, the plant can use some oxygen, right, during its aerobic respiration, but it doesn't need all of it. So it's going to also release some of the oxygen that it doesn't need during the gaseous exchange. Okay, so these are going to be some experiments that were used to prove certain factors are required for photosynthesis. Okay, so let's just go through this table really quickly. The basic knowledge: glucose is first formed from carbon dioxide during photosynthesis. Explanation: the simplest energy-rich organic molecules are carbohydrates, and the simplest stable form of carbohydrate is glucose. There is a presence of starch in the leaves, and that suggests that photosynthesis has taken place. When glucose is formed more quickly than it's used up, the excess glucose can be converted into starch for storage. This storage can happen in the leaves, or it can happen in other parts of the plant like the roots. Okay, and starch formation does not always indicate that photosynthesis has taken place, because starch can be formed even in the roots or underground storage organs of the plants. So we can actually test for starch using the iodine solution. Okay. So why this second point over here? Basically, this means that there could already have been some starch in the plant before the photosynthesis occurs. So what scientists usually do, they actually have to de-starch the plant to prove that whatever starch that it was shown in the iodine test, right, is like freshly made lah, right? It's new. Okay, so this starching must be carried out on the plant before experiments to ensure that the starch is absent in the leaves prior to experiments. Thus, all starch present in the leaves after the experiment must have been formed during experiments. So how to do that? We simply put the plant in the dark for 2 days, and then when it's in the dark, the photosynthesis stops, enzymes in the leaves will stop converting, will start to convert the starch into sucrose so that they can use the sucrose for other things like energy. Okay, so let's look at the first experiment, which is called the starch test. Okay, this one is simply to prove that photosynthesis has taken place. Okay, so this is a very simple experiment. Honestly, it can be done in about maybe 5 minutes or to 10 minutes. Okay, so you take a green leaf from a plant, right? So you break it off the branch. This plant needs to be exposed to light for at least a few hours, right? Ideally, maybe 5-6 hours. Immediately put the leaf into boiling water for 2 minutes to denature the enzymes in the leaf and stop the chemical reactions in the leaf. Okay, so you directly put the leaf into the boiling water over here. Okay, so this will denature the enzymes. Now you put the boiled leaf into a boiling tube that contains some alcohol. Okay, specifically, you can use ethanol that's very commonly found in the school lab. This will decolorize the leaf. Okay, and what does that mean? It means that the leaf will be the chlorophyll pigments in the leaf will be removed, and therefore you can see the leaf more clearly. Okay, so you know that chlorophyll is green lah, alright? So by removing these green pigments, now the leaf will become colorless / white. Okay, actually I prefer you to use the word colorless lah. Leaf become colorless. Okay, so the leaf is now very brittle. Okay, we need to put it back into the hot water. Okay, no more boiling water lah. Now it's just hot, maybe about 70-80 degrees Celsius. Okay, this will soften the leaf and make it more permeable to the iodine solution. Okay, because the iodine, it needs to permeate into the leaf cells. Okay, and therefore it also needs to pass through the cell membrane and all that, so you need to make it more permeable. Okay, lastly, you remove the leaf, you spread it evenly onto a white towel. Then you add a few drops of iodine solution onto the leaf. All right. So if there's a presence of a blue-black, okay, the iodine solution turns blue-black, right, from its yellowish-brown. Therefore, there's presence of starch, right? If the iodine solution remains yellowish-brown, starch is absent. Okay, so I've already explained all of this stuff. Okay, so you can just go to YouTube. There are plenty of videos where you can see this process taking place if you have not had the chance to do it yourself yet. Okay, one important thing, if you are doing this in the lab, never hold the tube of alcohol directly with a Bunsen burner flame, or never hit the tube of alcohol, because alcohol is flammable, and so it will definitely cause this whole tube to catch fire. Okay, so instead you put it into a boiling bath, right? A hot water bath or boiling water bath. Okay, one more thing about this is that you will notice that when you put alcohol into the water bath, even when you turn off the flame, as long as the water is hot enough, okay, as long as the water is hot, so when this process of decolorization is happening, make sure that the fire is off. You do not want the fire to be on. The water is hot enough, the boiling point of alcohol is lower than the boiling point of water, right? It's about 78. So as long as the hot water is above the boiling point of alcohol, that's good enough, right? It will cause the decolorization to happen. Okay. Okay, so next experiment. How do plants use light in photosynthesis? Again, this is a pretty common experiment. You just search YouTube, you'll see plenty of videos about this. Okay, so we start by destarching a potted plant, right, by placing in the dark for 2 days. Okay, so in this case, we destarch the entire plant, right? You put the whole plant in the dark for 2 days. Then you remove one leaf. You're going to test it for starch, as in the previous experiment that we just showed you. Now, you place the leaf, which is still attached to a plant. Okay, so you take another leaf, which is still attached to a plant, between 2 black pieces of paper. Okay, like this. Okay, so you just clip a black piece of paper, and in this paper, there's a cutout pattern. You can be any pattern, can be your name, can be a star, anything you want. Then you put this plant under strong sunlight. So after a few hours, you remove the second leaf, the one with the black paper, you decolorize it, and then you test it for starch. So you make a drawing. Okay, so what will you expect to see? If you make a drawing of this, the second leaf, right, then you are going to see something interesting. Firstly, the parts that are exposed to sunlight, like for example this part, the part of the star, the front part, these parts will turn blue-black, right? Whereas the parts that were covered by the black paper, they will not have any color change, right? You just remain yellowish brown, okay? But when you see on the leaf, actually it will just look colorless. Okay, so why do you test a leaf of starch before you even carry out this experiment, right? This is what I call as a control. You do not want the plant to, you want to make sure right? At step 2, you actually want to make sure that this plant has been properly destarched. Okay, so now we know so how do plants absorb light? Okay, chlorophyll is the pigment in plants that will absorb light, and the green color in leaves is due to the presence of chlorophyll. So do you know that there are some plants where they are actually variegated? Okay, so if you just look at this picture over here, you see that some plants are actually red, some plants like this plant over here is a mixture of green and white parts on the leaf. So a variegated plant is a plant with streaks and patches of different colors. But in our case, what we care about is that this variegated plant has streaks with chlorophyll and streaks of the plant leaf that does not contain chlorophyll. Right? So one example would be the Duranta species. So same thing, you put these plants in the dark for 2 days. After that, you expose the plant to strong light for a few hours. You remove one leaf. You start by making a drawing to show the distribution of the green parts so that you remember, right? I mean, you don't want to make drawing, you can always take a picture. Okay, and which parts contain chlorophyll, and which parts do not contain chlorophyll. Okay, then you decolorize the leaf, you test it for starch, and then finally you make a drawing to show the distribution of the blue-black color. Okay, so which part will you expect to see the blue-black color? Of course, it will be the part that contains the chlorophyll. The part that does not contain chlorophyll will remain yellowish brown. Okay, next experiment. Do plants use carbon dioxide in photosynthesis? I mean, we all know the answer to this, but how do we prove it? Here we have a very interesting experiment. Okay, so we start by destarching 2 potted plants and placing them in the dark for 2 days. Okay, so let's call this plant A and let's call this plant B. We enclose the pots in polythene bags. Okay, polythene is simply another word for plastic. So we secure the plastic bags to the plant stems. So some people will be like, what is this for? Okay, let me just tell you that this plastic bag is to prevent the gases or basically the gases that may come from the microorganisms that are found in the soil, right? We can't help it that soil always will have microorganisms, things like bacteria and other things like that. So we don't want any of the gases that are released from them. Okay, so we place 1 pot in the bell jar, and this plant does not have a supply of carbon dioxide from the air because soda lime and potassium hydroxide solution would rapidly absorb carbon dioxide. So at the top, we see here, there are beads of soda lime, and right beside the plant at the bottom of the bell jar, we have potassium hydroxide. So these 2 chemicals, they absorb carbon dioxide. Okay, so next we set up a control using pebbles and water, right, in place of soda lime and potassium hydroxide respectively, right? Of course, pebbles and water, they do not do anything, right? They will just allow the carbon dioxide to enter the bell jar normally. Okay, now we leave both set up in a strong light for a few hours. Finally, remove a leaf from each plant, test the leaf for starch. Okay, what will we expect to see? Okay, so obviously, this plant is not able in A, the experimental setup is not able to do photosynthesis because it doesn't have access to carbon dioxide. So if it cannot do photosynthesis, then that will mean that the leaf will not be blue-black, it will just remain yellowish brown. But for this plant, which is able to do photosynthesis, it will have some starch in the leaves, and therefore the part that have the starch will turn blue-black. Right, the iodine solution will turn blue-black. Okay, so conclusion, right? The conclusion is that over here, carbon dioxide is required for photosynthesis. Next, what gas is given off during photosynthesis? So, I mean, of course, we know it now. We know that carbon dioxide is absorbed, but oxygen is given off. But how did people prove this in the past? All right. So the experiment was like this. So we took some freshwater plants such as hydrilla, okay, and then you put it in this setup over here. You put the hydrilla below, and then you put a filter funnel above it, put a test tube that's filled with water, right, above it all. Here we have sodium hydrogen carbonate solution, right? This sodium hydrogen carbonate solution, right, it provides the carbon dioxide to the plant. Okay, you need might need quite a lot of it because you want to force photosynthesis to happen very quickly. Next, you place the setup in strong light for a few hours. You'll start to notice that gas bubbles will be forming on the leaves of this plant. And if you do it correctly, you need to kind of like trim this plant in the correct way, cut the stem of it in a very nice way such that the bubbles will be forced to move upward through the filter funnel, right? And then the bubbles basically the bubble of oxygen, right, will be able to move up into this test tube. It will displace the water, you'll push out the water, and then over time you start to see more and more oxygen start to enter the test tube over here. Okay, so once the tube is about half filled with gas, carefully pull out the test tube, cover it with your thumb quickly, do not let the gas escape. Okay, and then we are going to use the glowing splint test for oxygen. We light a small splint, we let it burn for a few seconds, and then we blow out the flame. So when the ember of the splint is still glowing, we place it back into this test tube gas sample, and if the glowing splint reignites, therefore you know that the gas is oxygen. Okay, so The answer is yes. As long as the photosynthesis occur, you will see the glowing spin reignites. Okay, so some discussion questions. Why is it important that no air? Again, is a control, right? Ensure that there's no the only gas that was trapped was the one that was being produced by the plants, and of course the rate of gas production, right? The more gas being produced, the higher the rate of gas production, the higher the rate of photosynthesis. Okay, let's talk about the chemical formulas. So the chemical formula for glucose is C6H12O6, right? Quite a simple way to remember, 612C, right? And again, you can remember Mr. Cho, right? CHO, that are the individual elements that make up this chemical. Okay, so how many carbon atoms does one molecule of glucose contain? 6. How many molecules of carbon dioxide will be needed to make one molecule of glucose in terms of the number of carbon atoms? So we see that one carbon dioxide molecule, right, or compound, it has basically only one carbon atom. So how many do we need? Okay, obviously answer is 6 also, right? So you will need 6 molecules of carbon dioxide. So what chemical element does a carbohydrate molecule contain that is not found in carbon dioxide? Suggest where a plant may obtain this element. We see that there's hydrogen also, right? There's 12 atoms of hydrogen, and so obviously the answer is water, right? H2O. So the water from the hydrogen from water will then be used to form the carbon dioxide. What am I saying? The glucose. Okay, sorry, still very early. I haven't wake up yet. Okay, let's talk about the conditions that are required for photosynthesis. Okay, we have shown that there's a need for light, there's a need for carbon dioxide, there's a need for chlorophyll. Okay, and so light, carbon dioxide, and chlorophyll are all essential for photosynthesis. But there are two other things that also are very essential. That is a suitable temperature, so the plant cannot be too cold, right? If not, the enzymes for photosynthesis will be inactive. It also cannot be too hot. If not, the enzymes for photosynthesis may become denatured, and the plant will start to also wilt, right? It start to become dehydrated, and it's not able to do photosynthesis. And of course, sufficient water, right? You need to water your plants. So photosynthesis depends on enzyme reactions in the chloroplast. So the effect of temperature has been discussed before in chapter 4. So we know that a suitable temperature is required for photosynthesis. So what happens during photosynthesis? So during photosynthesis is a process by which plants synthesize carbohydrates like glucose, specifically glucose at first, from water and carbon dioxide. Water and carbon dioxide are the raw materials for photosynthesis, and oxygen is released during the process. So during photosynthesis, chlorophyll absorbs energy from light and transfers it to chemical stores of energy in carbohydrates, for example glucose, which are synthesized from water and carbon dioxide. Oxygen is released as a byproduct. Here again, repeating myself many times. This is the equation that I told you before. This is the word equation at the top. And now we want to learn how to write a balanced chemical equation. Alright, so there's two kind of questions. One is that they ask you for word equation, then you write the word, and very simple. One is writing the balanced chemical equation. So the balanced chemical equation is 6 molecules of carbon dioxide plus 6 molecules of water, H2O, right, with light. Now light, of course, there's no chemical, and underneath the line, chlorophyll, will give you 1 molecule of glucose, C6H12O6, plus 6 molecules of oxygen. Now, actually, this is a simplified chemical equation. It's a bit more complicated than this, but that's okay, right? We probably don't need all this knowledge right now. If you're curious, you can ask ChatGPT, right, to explain the whole process, right? There's this thing called light dependent and light independent process. Okay, now let's talk about the rate of photosynthesis, right? It's very important to talk about how fast photosynthesis can occur. Yes, photosynthesis can occur, yes or no, but it's also a question of how fast or the rates of it happening. And it's affected by 3 external factors, namely light intensity. Intensity means the strength of something, the concentration of carbon dioxide, right, as well as the temperature. So let's look at these three effects or three factors. So we start to see this graph, right, where the rate of photosynthesis is on the y-axis, light intensity on the x-axis. So without enough light, a plant cannot photosynthesize very fast, even if there are plenty of water and carbon dioxide. So as you increase the light intensity, you increase the rate of photosynthesis until a constant rate is reached. Okay, so later I'll talk about this thing called limiting factors. So let me finish this whole section first. So when more light is applied to the plant, over here, more light is applied to the plant, the amount of energy, right, for photosynthesis increases. Therefore, you increase the rate of photosynthesis, right, in this section over here. Now, next thing, you can have plenty of light, but a plant cannot do photosynthesis if it has insufficient carbon dioxide. So by increasing the carbon dioxide concentration, you increase the rate of photosynthesis until a constant rate is being reached. When there is more carbon dioxide that's available to a plant, therefore it can produce more glucose. The last factor is temperature. So increasing the temperature to the optimal temperature will increase the rate of photosynthesis. Increasing the temperature beyond the optimal temperature decreases the rate of photosynthesis because of enzyme denaturation. Okay, you also remember that as you increase the temperature, you also increase the kinetic energy, more effective collisions, right, between the enzyme and the substrates, for more enzyme substrate complex. Okay, so we'll talk about how we can actually vary some of these factors. So the most common experiment for this chapter actually is this experiment over here. Okay, so unfortunately, if you weren't able to try it, again, go to YouTube. There are many videos on this experiment set up before. Okay, so what is the effect of varying light intensity on the rate of photosynthesis? Right, so we want to investigate the effect of different light intensities on the rate of photosynthesis. Okay, of course, there's some assumptions here. We assume that the rate of photosynthesis produced is directly proportional to the rate of oxygen is produced is directly proportional to the rate of photosynthesis, which is true, yes, but not always super accurate lah. But let's ignore that for now. Okay, so this is the procedure. We will set up the experiment as shown in this picture. Alright, so we will have a beaker with a thermometer set inside. We can even have a retort stand where we use the retort stand to hold up our either test tube or boiling tube which contains our plant. You might be wondering, how does this plant stay at the bottom? Right? Usually the plant might float up. You can use a simple stone or something heavy, tie it down and weigh it to the plant. Okay, you have sodium hydrogen carbonate solution in the boiling tube. That is to provide the plant with sufficient carbon dioxide. And you actually cut the end of the plant, right? So this cut end is actually facing upwards. This forces the oxygen that's being produced in the leaves to move through this stem and then release from the end of the plant, bubble up over here. Right? If not, the bubbles will come out from left, right, everywhere. So it's very hard for you to see the bubbles. Okay, then you have a electronically controlled water bath. Okay, so last time people always say, I need to put ice lah, I need to put hot water lah. Yes, that's true. It's like in our school we probably have to do that. Okay, but in exam you can just shortcut use the electronically controlled thermostat water bath to keep the temperature constant. Okay, then you have a lamp, right? Ideally you use a LED lamp. Don't use those kind of old school lamp which produce a lot of heat. Okay, because if you use those kind of lamp, then the heat might cause the temperature of the water to increase. So you use an LED lamp. The LED lamp usually doesn't more efficient, doesn't produce so much heat as a byproduct. Okay, so of course the wattage of the bulb must be consistent. Okay, so we realize that bubbles will be given off from the cut of the plant. you need to give some time for the plant to adapt to the conditions before you take the reading. So every time you set this experiment up, or even when you move the lamp back and forth, you change the distance of the lamp or whatever, right? You always need to let the plant get used to the condition, adapt to the condition. So maybe give it about 3 minutes, I would say about 3 minutes before we can actually start to count the bubbles. So once we see that the bubbles are being produced at a regular rate, so it's bubbling, 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 we can then count the number of bubbles over a period of 5 minutes. So all we need to do is sit there with our stopwatch for 5 minutes, count our bubbles, and then you can repeat each step a few times, right? Repeat each at each distance. You repeat a few times, I'll say 3 times, right, to find the average rates. Okay, so what do you mean by rate? Rate is a measure of the number of bubbles over a certain period of time. Okay, so for example, 80 bubbles over 5 minutes, right? That means that how many? Right, about 18 divided by 5, oh my gosh, 16, right? Okay, it's 16 per minute. Okay, that's the average rate. Okay, next, we need to repeat the steps, right? Step 3, by changing the light source, the distance of the light source. Okay, so for example, we need 5 independent variables, right? In this case, the independent variable is the light intensity. The light intensity is controlled by the distance of the lamp to the plants. Okay, so for example, this lamp, right, you can put it at 80 cm, then you can change it to 40 cm, 30 cm, 20 cm, 15 cm, and 10 cm. Okay, now I don't really like this kind of inconsistent scale, so it'll be, in fact, personally, what I'll choose is I'll put like 50, 40, 30, 20, like that. Okay, I need 5, so maybe 60, 50, 40, 30, 20, 10. It cannot be 0 lah, cannot be the 0, impossible, right? That means the lamp is in the water already. Okay, so the nearer the light source is to the bigger, the higher the light intensity that the plant will be exposed to. Okay, you can record your results in a table, plot a graph to show the rate of bubbling per minute against the distance between the lamp and the plant. Okay, so from our investigation, we can conclude a few things. The higher the light intensity, okay, the higher light intensity, the higher the rate of photosynthesis based on the rate that there are more air bubbles being given off. Okay, so that's a slight assumption, but that's okay. You don't need to write that part in your answer. Let's just say it's a fair assumption. Okay, this one I already told you. Sodium hydroxide is to help to provide carbon dioxide to the plant. And then what are some things that actually keep constant, your constant variables, things that you must actually write in the exam when you're planning? Things like, firstly, the temperature, right? The temperature of the water bath must be kept constant, and things like the concentration of sodium hydrogen carbonate solution must also be kept constant. Okay, of course, if you can keep things like constant, like the plant that's being used, right, the size of the plant, okay, the number of leaves on the plant, those are things that you need to keep constant. Also, make sure that you remove any external sources of light. For example, if you are using this experiment, you must turn off the lights in the room, right? You must close the room, darken the room, close the curtains, everything, right? So that the only light source comes from the lamp. Okay, so the next 2 experiments will be very, very similar to what I just said. It's just that we change a few things. We're going to investigate the effect of different temperatures on the rate of photosynthesis. Okay, and how do we do that? We're going to use the similar experiment. This time, we just place the lamp 10 cm away from the plant. And we're going to keep this distance constant throughout the whole investigation. We will add ice cold water, right, to the water bath and keep the temperature at 5 degrees. We then allow the plant to adapt to this condition, so they about 3 minutes. After that, once the plant is adapted, we count the number of bubbles over a period of 5 minutes, repeat it to find the average rates. Okay, so then we repeat step 4 with different temperatures. For example, 50, 25, 35, 45, 55, 65, and 75. How do you do this? Of course, it's by using a mixture of hot and cold water as well as ice. Okay, if you are very lazy to write all this in the exam, what I can say, you use a electronically controlled thermostat water bath, right? Then you can just set the temperature to whatever temperature that you want. Then the thermostat electronically controlled thermostat water will just change the temperature for you. Okay, now for each temperature, you need to use new sodium hydrogen carbonate solution. Of course, remember to keep this constant, keep the concentration constant. What other things do you need to keep constant, right? The distance of the lamp, you also need to keep it constant. Don't go and suddenly change it for some reason. Okay, blah, 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 blah. What will be the effect of temperature from 5 to 35? Okay, so assuming that this plant has an optimal temperature of 35, okay, because every plant has their own optimal temperature, then you'll see the rates of photosynthesis start to increase from 5 to 35 because it's from inactive, right, enzymes inactive to its optimum temperature. At which temperature will be the rate of bubbles given off the highest. This will be the optimal temperature. So what happens if the temperature is increased beyond the optimal temperature, right? Then the enzymes required for photosynthesis will start to denature. The rate of there'll be less bubbles being produced, so the rate of bubbling will reduce, and therefore the correlation is the rate of photosynthesis will also be decreasing. Okay, next thing, what is the effect of varying carbon dioxide concentration on the rate of photosynthesis? We do the exact same experiment. We put a lamp 10 cm away from the plant. We keep the distance constant throughout the investigation. We conduct the investigation at room temperature. Okay, so this one we don't need to do anything, just put it at room temperature. Make sure that room temperature it stays constant also. So in fact you can say conduct the investigation at a constant room temperature. We use different concentrations of sodium hydrogen carbonate solution, for example 0.01 mole per dm minus 3, blah 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 blah. Don't say all of this. Okay, but when you write the exam, you need to write it out like this lah. At least 5 different independent variables. I want to say 5 points lah, right? For your independent variable, not 5 different independent variables. Okay, so so this will be proportional to the carbon dioxide concentration in the solution. So what again, let the plant adapt to the condition. Once the bubbles start to come at a regular rate, you measure the rate of bubbles per minute for each concentration of the sodium hydrogen carbonate solution. Record the results in the table, plot the graph, show number of bubbles per minute, blah, blah, blah. Okay, what does this show? The higher the concentration of sodium hydrogen carbonate solution, the higher the rate of photosynthesis, because there is more carbon dioxide available for the plant to use. Okay, so I'm going to pause here. I think I've said a lot already, and so we'll continue this lecture series over the next few weeks. Okay, so I know that it's a long series, but right, for those that were not in class at a certain point of time, I hope that this lecture series will help you a lot. Now do feel free to watch this video again, watch it, if you need to watch it at faster speed, slower speed, it's up to you, but as long as you are able to learn properly this chapter, and if you still cannot get something, the experiments are a bit tricky for you, no worries, just come and talk to me, alright? Next thing you can do, watch the videos for all of these experiments. There's plenty of them out there. All you do is to search this head bar over, just search effect of temperature on rate of photosynthesis, but you'll get 100 videos for you to see. Okay, with that, I'll end this lecture for now, and then I'll see you back in school. Thank you, bye bye.